Right, thanks very much. So I'm going to really lead on from where we've heard from Tina, from Sami and Olivia, who've done a lot of the heavy lifting so far, and I'm going to try and tie it a little bit together and to look a bit further into the future as to new techniques and how we might use the models and the data to understand what's coming down the line. So, as we've seen, Sami and Olivia are only 2 years into their PhD, but I think they're making really astounding progress. They've moved very fast with the data and the models, and the next stages now are going to be where they start to integrate the data, the models in an iterative process, and use that to move forward in a more integrated way. But we have seen already some of the first publications are now actually in press and starting to appear, even at this early stage. So in this talk, I'm going to cover a few key points. So I'm going to consider salmon as keystone species in the food web, why we need to use this food web approach. Think a bit more about the 6 rivers as a model system to understand fundamentals that are important here, but also further afield. And then to think about this kind of bigger picture and also projecting into the future, and considering particularly both threats and opportunities on the horizon. So one of the key things really at the start of this project was kind of identifying bottlenecks and then thinking about how we might move forward. So enriching existing data with new approaches that haven't really been used so widely, at least from the perspective of salmon. So luckily for us as sort of general ecologists, salmon are among the most heavily, intensively studied species anywhere on the planet, particularly for vertebrates. But really, despite this huge wealth of data, much of the focus has really been on salmon, only with very little consideration of the food web and the wider ecosystem within which it operates. So salmon affect the food web, and they are also affected by it, and they have ecosystem consequences. There's also been an issue of a sort of general lack of data sharing, often due to institutional issues and mechanisms for spreading the data around and using it more effectively. And I think Colin Bull's going to talk a little bit more about that. So there are initiatives that are opening up the use of data to make it more effective for us. There's also been rather patchy life cycle coverage, and as Tina has already said, there is this big black box around the marine phase. So what we need to do is to join the dots. We need to think about how salmon operate over space and time and how they move through the food web, so moving beyond the population level to look down the microscope to the individuals and the components of the life cycle, and through the telescope to look through the ecosystem. And really we're standing on the shoulders of giants here because many people have done hard work collecting all this data over a long time, and we can make strong advantage of that by then coming in and layering these new approaches. And just to touch on why we're using some of these approaches, what we need to do is to understand some of the fundamental architecture of life in these systems and what is constraining the salmon and the species around them. And as we've already seen hinted at in the other talks, metabolism is really key. And metabolism of life on Earth, of all species, it scales with body mass, and it follows this general relationship, a power law, where we have a logarithm of metabolic rate on the y-axis and a logarithm of individual body mass on the x. And what we can see here is everything from algae down in the bottom left to the largest thing on the planet, in fact the largest thing that has ever lived, the blue whale at the top right, they fall on this general line. And then if we think about the 6 rivers and many other systems that hold salmon, they fall in this sort of red box here. So this is the architecture really and the backbone of ecological energetics, that we start to go from the individual through to the population and to these higher organizational levels. But thinking about the implications of this for salmon in particular and the different components of the life cycle, you've seen in the previous talk from Olivia, we have these thermal performance curves for all species on the planet, and salmon are no exception, where there are optimum temperatures with an ascending limb where performance increases with rising temperature, and then a descending limb where performance starts to be impaired. And what we can see here in particular is that in the salmon life cycle, they all follow these unimodal relationships, but the peak of the curve and the steepness of the curves differs depending on where you are in the life cycle. So in general, from this meta-analysis that Olivia has conducted, what we can see is that fry generally have a colder optimum than parr, for instance. And these are going to be important when we start feeding them into these matrix projection models. And we can also take some of these individual-based approaches and then think about them actually in the field. So these are some field data that we collected a few years ago with Owen O'Gorman, a colleague of mine, where we measured the respiration rate, which is a measure of the individual metabolic rate of salmon in rivers in Iceland. Some of these are close to Reykjavik in the Hengith catchment, but also a range of the 6 rivers. And we measured this for both salmon and trout. And what you can see here is as body mass increases, the respiration rate rises, so this metabolic rate rises, but there are different slopes for the salmon and the trout. And this could be quite important when we start thinking about the 2 species together, how they coexist, and why salmon might need to leave the river, for instance. What seems to happen here is that although at small body sizes, they're pretty close in terms of their respiration rates, as they get bigger, their metabolic rate and therefore the amount of food they need to eat to meet those demands starts rising much faster for the salmon. It becomes more challenging for them to stay in the river as they get bigger relative to trout. And we also see that these individual relationships with body mass and temperature, etc., and metabolism, they also are really important when we start thinking at the higher organizational levels and what the ecosystem is doing. So this is an example from a review I published in 2005, where we started looking at things like home range, nutrient turnover, feeding rates, and all of these different ecological processes and how they then scale with body mass. So we have these power law relationships are pretty strong when we look across ecosystems, and they again help us provide this backbone, this context for trying to understand what happens when systems start deviating from these underlying ecological rules. These are some more data that are more recent from surveys that we've done through the literature and samples collected recently in the 6 rivers and also from the marine literature, where we started to assemble the food web for the salmon to try and understand how it sits in these trophic networks. In this diagram, what we've done is for Icelandic marine webs and freshwater webs, we've put them into these circular networks. Each dot, which is tiny in each one of these, is a different species. So here we're dealing with over 100 species in the marine system and still a relatively large number in the fresh waters. And we've put salmon at the center, so the red dot in the middle is the salmon, and those species in the circle next to it are separated by 1 trophic link, either as predators and prey. Those in the second circle are separated by 2 trophic links, and those in the third circle by 3. Essentially, what we can see is even in these really complex marine webs, salmon are no more than 3 trophic links away from any other species. So they're highly connected, embedded within the food web as predators or prey. And the same thing for the freshwater webs, but here they are no more than 2 links away, so even more closely interlinked. So that's why these food web approaches are important. We can also think about the 6 rivers as a model system and the strengths that they bring. So some of these things we've already been touched on. So they have an exceptional time series on the long-term salmon populations. Now we're trying to enrich that with these new levels of data. Iceland can also be thought of really as an ark for the salmon in terms of things like climate change and the amount of pollution, etc., the threats that they face here. This is really the kind of stronghold that salmon are going to be holding out probably later than anywhere else. And they have these relatively simple food webs. So all the food webs you saw look complicated. They are much less complex than in other systems. So this is an ideal way where we can start to unpick cause and effect in these systems and to understand how external stressors here and elsewhere might start operating on salmon dynamics. And then the final point here as well is this ability to use before and after control impact designs of management actions. So here we can look at the reforestation, as Pete touched on, salmon ladders, egg planting, catch and release, these other actions that Helena mentioned, and test these in a more controlled way. And we can also use space for time substitution by looking at sites across different spatial gradients, for instance, from Iceland further south, to try and understand what might happen into the future as the systems here maybe start to warm, for example. This is just an example of some of the things that we can do when we have these food web data. Here we've taken the food webs and collapsed them into these more simplified trophic biomass pyramids, where what we have is on the bottom, the green blobs are algae, the base of the food web. We have these brownish blobs, which are the invertebrates above that, and then we have the fish that eat invertebrates shown as the red blobs. Now, there's a range of the 6 Icelandic rivers shown here to the left, and then we have some UK rivers for comparison over to the right. And what you'll notice is that there's a generally fairly consistent pattern across here in terms of how the biomass is distributed across these trophic levels. But notice in the UK, over to the far right, the Froome, which Rasmus Lauridsen will speak about later on in the meeting, we have an additional trophic level where we have piscivorous fish such as pike perched on top of the salmon and the other fish. But a key point to notice in terms of the Icelandic rivers is this blob here where we have missing salmon highlighted, where the red blob is constrained. It's not as wide as in the other systems. And here, what we find, this is in the Mithi, which is the river where there is a more recent salmon ladder that Hlina mentioned. And above that ladder, we sampled that system, and essentially, we're looking at char and an absence of salmon. So there are salmonids, but not salmon. So the difference between that blob and all the other ones is suggestive to us that there is headroom, extra space for the salmon to occupy this biomass at the top of the web. So this can be tested experimentally by the work that Sami will be doing later. Okay, so that's just a taste of some of the things we're doing in the early stages. I'll mention quickly now a little bit of what we might need to start thinking about in terms of the longer-term issues facing the salmon here. And just as an example of thinking about one of these species, which Hlina has mentioned, this massive exponential rise in the abundance of Pacific salmon, we know that these are really important keystone species in their native range. Just as Atlantic salmon are important here in the Pacific, these species play an important role. They deliver marine-derived nutrients to nutrient-poor systems. That fuels the food web, and it also kickstarts the next generation of the juvenile salmon. And the adults themselves are eaten by bears. We all know what bears go and do in the woods. They transport the marine-derived nutrients, and they stimulate forest growth by moving this marine subsidy into the terrestrial zone, which then actually feeds back into the rivers. So we need to think about what the Pacific salmon might do. In reality, eradication is likely to be impossible given the scale of the challenge now. The sheer numbers and spread of these things means that they are something like the SARS-CoV-2 virus we're going to have to learn to live with. But we need to understand, under this new normal, what can we do? How do we protect the Atlantic salmon within this new normal, as it were? And the new data and models that we're working on here are going to be essential. But now is the time, this window of opportunity, to establish the baselines, to understand what's happening, and then to try and anticipate what we might need to do to act in the future. So will pink salmon fertilize riverine food webs with marine subsidies, as in North America? It could be that they actually trigger in-stream productivity to higher levels. But they could increase competition with Atlantic salmon in the marine phase, especially if they hang around the coast, which certainly it seems to be the case. That could mean that Atlantic salmon have to run a gauntlet in this stage where we're already seeing potentially high mortality in that early marine phase. And then the question is, can we nudge the systems to protect the native species? Another issue that we need to be aware of in Iceland in particular is the invasion of these lupins, which are spreading across the landscape at a rapid rate. So these are changing the base of the food web in the terrestrial zone. And we see that this is actually very important in the freshwater food webs and has implications for salmonids, because these are nitrogen fixers. They improve the fertility of the soil. These nutrients can eventually leach out into the waters, improving algal productivity, invertebrate productivity, and potentially more food for the fish. And they could also introduce terrestrial prey such as pollinators as a direct new food subsidy for the fish in the rivers. And we know that although chironomids are the dominant aquatic prey of the salmon, they are opportunistic and they'll take terrestrial prey. This is an example of salmon guts taken from the Vestadalsa. You can see these earthworms which were collected after a storm event. These are washed off from the terrestrial zone into salmon, and the equivalent amount of biomass there, if you were making that worm biomass from chironomids, you would probably be looking at about 10,000 midges to get the same amount of protein. So they will interact in the terrestrials with the terrestrial subsidies. What we can predict then is that the increased terrestrial productivity that we're likely to see through reforestation programs such as being trialed now and instigated in the 6 rivers is likely to add fuel to the base of the food web. That will benefit the trout probably the most of all because they have a stronger link to terrestrial subsidies, but also salmon too. And what we predict there is a rising tide here will raise all the boats. We also know that the leaf litter that leaves these forested systems in the future is processed by species such as caddisfly, which Hlina mentioned are present in some of the rivers here. These feed on leaf litter, but the leaf litter food resource has been absent for much of our Icelandic streams for a long time due to the clearance of the forests. But it means that when the forests regrow, this part of the food web can be activated and it produces this new food source for the salmon in the future. And then just to finally finish off, we're going to think a little bit now about some new tools that we might be able to use to improve or at least speed up the data generation. So Sami's work in particular is doing stuff that I spent a lot of time doing, identifying invertebrates down a microscope, which is extremely time-consuming and very slow. The question is, can we use molecular tools to do this faster but without sacrificing accuracy? And can we also use that to track native salmon, as well as Pacific invaders? So this touches on this eDNA revolution, this environmental DNA revolution, where we can take DNA from the water, we can sequence it, and now we can use that to give us a complete biodiversity inventory of every species in that system. We can do this for terrestrial systems and fresh water, where you can get complete coverage from microbes to salmon and everything above them, with a small sample and relatively low cost now. We can also quantify focal species using this quantitative PCR, which all of us are now familiar with because we all went through that to come here through our qPCR tests. It's exactly the same approach, and we can use that to look for Pacific salmon or spawning events in the rivers. So the idea here essentially is we can take water and sediment samples and sequence it, and that will tell us what is in that river. And the 6 rivers here is really ideal because we can calibrate that against the real traditional approaches using empirical data to understand how effective it is. And this is what the process looks like. We take these samples from the river, we run them through sieves, centrifuge them, and sequence them to tell us what species are present. This is a process called meta-barcoding, and we've done this recently in a paper where we did it in a fraction of the time and a fraction of the cost for what we would do with using our traditional techniques. So these eDNA and qPCR approaches, they are incredibly sensitive. Just as an example, a recent study in Wales in the United Kingdom, a small bag of mackerel was placed in a lake, and those mackerel could be detected many kilometers downstream of the outflow of the lake. So here we're talking about literally picking up molecules in a huge volume of water. They're incredibly powerful. Okay, so that's some of the new things we want to do. The next steps on the near horizon now for Sami and Olivia are to work with integrating the data and the models, to link that through to management actions as well, and then to move from these general models that Olivia has been building to then to start tailoring them to more specific systems, such as the Vesta Dausa, where we have the greatest data. That will be the next starting point. So I'd just like to say watch this space as they move through the PhDs. Thanks very much.